Hello and welcome, everyone. My name is John Kalantari. I'm an assistant professor in the Department of Surgery and Quantitative Health Sciences and faculty member in the Center for Individualized Medicine at Mayo Clinic. And I'd like to welcome all of you to our Advancing Care Through Genomics virtual speaker series focused on AI and machine learning. And today is our second presentation in our multi-part series. Dr. Khezeli and Dr. Nicholas Chia are joining me today as co-moderators for the event. Dr. Chia is the director of Beyond DNA and co-director of the microbiome program within the Center for Individualized Medicine. And Dr. Khezeli is a machine learning scientist in the Biomedical Artificial General Intelligence Lab at Mayo Clinic. Now, before we get started, a few housekeeping items. Please use the Q&A in Zoom to submit your questions. We do have time planned for a Q&A at the end of the session. And for those of you interested in earning continuing education credit, an email will be sent within 48 hours that includes instructions on how to claim credit. And with that said, today we're delighted to welcome Hossein Sharifi Noghabi as our featured speaker. Hossein is a doctoral researcher in computer science at Simon Fraser University and affiliated with the Vancouver Prostate Center. He received his bachelor's in information technology and master's in artificial intelligence 2012 and 2015. And Hossein's research is on transfer learning and out of distribution generalization in drug response prediction and pharmacogenomics. With that said, welcome, Hossein. Thank you for joining us today.
Thank you so much, John. Thank you so much for having me today and for the introduction. I'm super excited to talk about part of our effort on transfer learning and drug response prediction for you. So let me share my screen. And hopefully now you can see that. So today, I'm going to talk about transfer learning and drug response prediction and connect these two areas that seem to be distantly related, but they have very, very close interactions with each other. So probably it's general knowledge for most of you, but to feel good that I'm starting at the right position, the whole premise of precision oncology is to provide individualized treatment for cancer patients based on some omics features that we can get from these patients. But the problem is that currently limited number of patients are benefiting from this because cancer is a very heterogeneous and complex disease. And unfortunately, for many privacy or ethical considerations, this clinical data is not publicly available. Plus, it's impossible to study the impacts of multiple drugs separately or in combination. So to solve these problems, a lot of focus has been on preclinical resources, such as patient-derived xenografts, animal models, cancer cell lines, and most recently, organoids. And we can get much larger sample size for these preclinical studies. We can screen them with hundreds of drugs separately or in combination, sometimes less ethical considerations, and often multiomics data for each sample, which is great. And that's why a lot of focus have been on developing these pharmacogenomics studies, starting with NCI-60 cell lines back in the 80s, that later evolved into different consortiums such as CGP at Sanger, CCOE at Novartis, PDX Encyclopedia, and other data sets that you see some characteristics of them down here. A lot of experiments, a lot of sample size, screens with hundreds of drugs, and we get multiomics data for the majority of them, which is awesome. Just a quick overview of where this data is coming from. We usually start with a biopsy from cancer patients, and those cancer cells go through multiple factors to immortalize them and prepare them for expansion. And then we can store these cell lines and distribute them to different labs if they want to. And we can obtain different omics data type of molecular profiles from these cancer cell lines and use them in any analysis. And it's a similar story for patient-derived xenograft. Start with a tumor implanted in mice, and we can pass it to the different generations and obtain molecular profiles at each stage. And then we can design the treatment and control group and measure the response to a particular drug. This response is generally measured differently between cell lines and PDXs. In cell lines, we work on concentration of the drug over time and measure the viability. and often refer different statistics from those response measurements. The most common one is IC50, the concentration of the drug that kills half of the cell, reduces viability by 50%, or we can get something like Emax, which shows the maximum amount of response that we can get. IC50 is a measure of potency and Emax is a measure of efficacy. We can summarize them into one number by focusing on area above this curve, also known as AAC, or area under this curve, known as AUC. But we can get these measures based on changing concentration in PDXs, but instead measures based on changing the size of tumor after treatment are used. One of the most well-known one is, for example, RECIST, that categorize the response into 4 categories of complete response, partial response, stable disease, and progressive disease. Now that we have the molecular profile as an input and we have the measure of drug response, either in form of IC50, for example, RECIST, we can design a computational method that predicts drug response. Ideally, we want to build these methods for patients, but as I said, because of the reasons like a lack of enough sample, we can't really do that. So we go to next best things and try to develop these methods on preclinical resources, such as PDXs or cell lines. And we hope that such model generalizes to patients. But there are many challenges for drug response prediction. Obviously, the most important one is that we want it to be as accurate as possible. If we have multiomics characterization of the preclinical samples, we want to use all the information that we have, but integrating these different data types, it's not easy. More importantly, a bunch of cancer cells on a plastic dish are obviously very different than the complex nature of human body, lack of tumor microenvironment, lack of immune system, you name it. But we need to be able to address these discrepancies if we want to use all these resources together. And at the end of the day, we need to be able to interpret and explain the predictions that these computational tools are making, which is not easy at all. So a lot of, the focus of my talk today is mostly around the third point, but I'll touch upon all of these hopefully today. A lot of methods have been developed in the past couple of years to address one or some of these challenges. A quick overview of this figure. This is not an exhaustive list, it's a cherry-picked list. We see that, for example, gene expression in blue diamond is the most common input data to build these computational methods to predict drug response. And the most common measure of drug response to build this model is IC50, but we see a shift in the recent years towards using AUC or AAC area above or under dose response curve instead of IC50. A look into the data set also tells us that GDSCV1 from Sanger Institute is the most common data set for training these models, but in recent years, CCLE and CTRPV2 are gaining some interest, and also using additional information of the drug, such as structure of the chemical compound, and things like SMARS representation, Morgan fingerprint is also getting common to build these models. The majority of these studies focused on deep neural networks or deep learning because like many areas, this new line of research revolutionized computational biology and bioinformatics as well. And they're showing interesting results in genomics, biology, and medicine. So today, whenever I talk about a model or a predictor, I'm referring to a deep neural network. But what does this network do? A deep neural network performs a layer-wise transformation of the data. Imagine you have a gene expression matrix of sample and some genes. A deep neural network at the input layer receives this raw data, extracts some features from it, and pass those features to the next layer. Each layer extracts more abstract features from this input and pass it to the next layer until you reach to a point that you get a very rich representation of the original data using these extracted features for the same samples, but in much lower dimension. And you can use these rich extracted features for any downstream task. For example, in our case, we want to add a predictor or classifier or head. There are many names for this part. We can add this part to make predictions, accurate drug response prediction. And the way these networks work is that they compared what they predict to some ground truths that we get from the wet lab or some other measures. And based on how much we are deviating from these ground truths using a loss function that calculates the error that we have, we send back the error that these feature extractor and predictor make back to the network to optimize their parameters. All these lines that you see connect these computational units are different parameters that these models have that map this raw gene expression to ultimate predictions. And when we send this correction signal back to the network and optimize everything, hopefully we get a more accurate prediction. So we do another forward pass to get the new set of predictions. And we repeat this process, which is called training, a lot of times to make sure that this model is as accurate as possible. And when we are happy with a trained model, we perform one step of test using a completely new set of data, withhold data that we don't have access during training, and test the model once and get the prediction and see how good we are in making accurate drug response prediction. Generally, we have two options to build these models. We can either build them within domain or we can build them in cross domain. Within domain, we train and test on the same type of data. For example, if we're training on cell line A, we test it on the same cell line with the withhold part, which usually, for example, you can do it using cross-validation or something like that. Or if you're training on cell line A, you test it on cell line B. What I'm trying to say is that we stay in the realm of the same data that we train the model with. But if the ultimate goal is to use this model in the clinic, you want to test them on patient data. So you need to go to the cross domain as well, which means that you train the model on cell lines or some other preclinical data, but test it on clinical data, which is great. You can study the translation to the clinic of what you design. But the caveat is that there are also sort of discrepancy and inconsistencies between these data sets. For example, gene expression data is different between cell lines and patients for many obvious reasons. As I explained at the beginning, even measures of the drug response are different between cell lines and patients. In cell lines, we have IC50, but in patients, we can get response based on changing the size of tumor or concentration of a protein in blood. There's even a huge line of discussion in the community that two cell line data set can have inconsistencies and discrepancies. So how can we make accurate drug response prediction using these computational tools while our materials, our input data have all sort of inconsistencies and discrepancies? To address this, we have two ways. One line of effort is try to gather and integrate data in a more standardized way. For example, PharmacoDB and CellMiner CDB are examples of such projects that they try to provide the community with data sets that are more integrated and standardized. The other line of work is to design computational tools that can handle data discrepancy when data is coming from multiple resources. And this line of work is based on transfer learning and the focus of my talk today. So let me motivate transfer learning by an example. Suppose you have a very data set of very nice sketches, professionally drawn art, and your goal is to build a model to classify them accurately in these categories that you see. Unfortunately, because getting this nice sketch is very hard, you don't have enough samples to do that. And you're not a rich PI like John. You cannot hire professional artists to do that for you. So what are you going to do to solve this problem? Well, one option is that to step up and draw them yourself. But I think you agree with me that this is a disaster. This is not what we are looking for. Another option is that you can go over the internet and try to find as many images as you can to solve your problem. And this is good. Now you have enough sample to build your computational method to classify these images. But the problem is that if you use all these images to build a model to predict these nice sketches, instead of targeting for those sketches, you're aiming at the wrong direction. The reason is that obviously the data that you're using to build your model is very different than the data that you eventually want to test it on. Ideally, you want these data sets to be similar to each other, but a close look at any of these categories tells you that they're very different than the data that you ultimately is going to test the model on. You may get some very, very bare hits, but this is not what you're building this model. So we need to be able to use all of these resources in a right way together. Transfer learning attempts to solve this problem in a very delicate way. In traditional machine learning, we have separate models for every data set that we may encounter and the different tasks that those data sets have, classifying circles or rectangles and so on. But in transfer learning, our goal is to build one learning system for the task that we care about, the target task, using the knowledge that we can get from some other relevant tasks known as source tasks or source domains. And the idea is that these source domains are going to help us to build a more accurate model for the target. The target is the thing that we care about and we are, the reason that we are going through all this trouble to build this model. In this example that I mentioned for images, the source domain is all those images that we could gather, and the target domain is those nice sketches that we cared about. And our task here, our target task here, is to classify these images as accurate as possible to these categories. And this is the exact scenario that we have for drug response prediction. Ultimately, we want to build an accurate model for patients, but we don't have enough patient data. That's why we go and gather as many cell lines as we can to help us to build the accurate model for eventually for patients. Now, transfer learning can be categorized into unsupervised, transductive, and inductive. Drug response prediction is a case of inductive transfer learning because we have different domains. By domains, I mean we have cell lines as source domain and patients or PDX, for example, in the target domain, which are obviously different. We also have different tasks. We want to achieve different goals on them. Of course, they're both related to drug response prediction, but for cell lines, we are predicting IC50, a continuous measure of drug response. But in patients, we are predicting RECIST, a categorical measure of drug response. So in one side, we have a regression problem, and the other side, we have a classification problem. And these two tasks are different. But we do have access to label in both source and target. We have IC50 in cell line as the label, and we have RECIST as the label in the target for patients, which is good. But the challenge for us is that the gene expression, the input side of this problem is different between cell lines and patient, and the measure of the drug response is also different between cell lines and patient. So drug response prediction has this unique challenge that we have both input and output spaces discrepancies. So how can we solve this? Ideally, our goal is to have a learning system that can adapt both input and output by taking gene expression from cell lines and patients and making accurate predictions in terms of both IC50 and RECIST, which again, we have two different tasks in drug response prediction for cell lines and patient. And the hypothesis here is that if we be able to adapt both input and output, we should be have a more accurate model compared to only adapting one side of the story, for example, only adapting the gene expression or only adapting the measure of drug response, or ignoring these data discrepancies completely and assume that these data sets are the same, which we know they're not. But unfortunately, we don't have any method that can perform this adaptation at both the input and the output side for us. So my goal here today is to design such a method. Let's focus on our problems one at a time. Let's, for now, assume that we only have output space discrepancy. We have different measures of drug response between cell lines and patients. So this is what we have. We have gene expression for some genes in patients, and we have gene expression for similar genes in cell lines. We have IC50 for cell lines, and we have RECIST in patients. We know that we can build a feature extractor, as I explained earlier, and extract rich features from both of them. Ideally, we want two different predictors, one for cell lines and one for patient. I call this predictor because it's a regression. It tries to predict IC50, a continuous number, and call this classifier because it aims at predicting RECIST, a categorical measure of drug response. This is ideally what we want to have, but unfortunately, we can't do that because our patient data set is very small, and we can't afford to train a completely separate classifier for patients. So let me be sneaky a little bit and merge these two into one predictor. This predictor has a classification part specifically designed for patients and a regression part specifically designed for cell lines. They also share some part of the network at the beginning because, as I said, drug response prediction in cell lines and patient, they are related to each other. So it makes sense that we use one predictor for both of them, but also respect the differences that they have in terms of measure of drug response. So if we use this merged network and train drug response prediction simultaneously for both cell lines and patient, we can achieve our goal and have the labels that we are looking for, making accurate predictions for both of them at the same time. And the beauty of this approach is that I can apply the classification part on cell lines and get the categorical label for cell lines, because I don't have that. I only have IC50 for cell lines, but I need both of them. Similarly, I can apply the regression part on patients and get the IC50, the continuous measure of drug response for patients, because I don't have that, but I may need it. This way, we are making accurate predictions for both sides. And at the same time, we can assign the label that we didn't have for them in the original data by applying that specific part to the data that we want. And this is by no magic. It's been known as multitask learning in computer science for a long time. The idea of multitask learning is training multiple related tasks together when we know that they can benefit from each other. One famous example is in autonomous driving, because detecting a traffic light is very related to detecting a pedestrian or road features, and they all can help each other. And this is a similar story here. Our goal is to train these related tasks together such that task C benefits from the data that task A and B have. And similarly, task A benefits from the data that task B and C have. The way these networks, multitask networks work at the most simplest scenario is that they have some shared part that is trained using the data of all these tasks, and some specific part that is only trained using the specific data for that corresponding task. And this is exactly what we did to solve our output problem. We used the gene expression of cell lines and patient as input and predicted drug response for each of them simultaneously, but respected the difference between them as well. So this seems to solve our output space problem. But I still owe you to solve the input problem because, as I mentioned before, gene expression between cell lines and patient is different. And when we extract features from cell lines and patients, this is what is going to happen. Imagine the green one is patient and the blue one is cell lines. And we have some responders that I show by cross and some non-responders that I show by plus. Ideally, we want all the non-responders be on the right side and all the responders to be on the left side. But because these two data sets are different, gene expression between cell lines and patients are different, we get such extracted features from them that they are not aligned. We may get some accurate predictions using the multitask network that we designed. Imagine this black line is what multitask part is doing for us. But we can't be as accurate as we want because we didn't respect the differences that they had at the input side. When we have the raw input, we can't do anything about the gene expression because that's what we measured. But what we can do is to train our network to extract similar features from both patients and cell lines. In the raw space, if they are here, we want to extract features, map them to a space that they are more similar to each other and closer to each other. But the question is, how do we want to do that? For that, let me deviate a little bit and go to computer vision and introduce generative adversarial networks for you. A generative adversarial network, also known as GAN, has a two network that compete with each other, a generator and a discriminator. The generator receives nothing but random noise and creates a fake sample, fake image. The discriminator receives a real sample and a fake sample and should be able to classify it accurately. These two networks compete with each other because they have different objectives. The discriminator wants to maximize its accuracy of catching real and fake, while the generator wants to minimize its error of being caught by the discriminator. So when these two networks constantly compete with each other in the process of training, we get more and more better fake images until we reach to a point that ideally discriminator says there's a 50% chance that this sample is fake and 50% chance that this sample is real. And the benefit of that is doing that is that the generator learns the characteristics and the distribution of the data that we have. At this point, you may wonder, how is that relevant to our problem in adapting the input space, the gene expression between cell lines and patients? We can use the same idea for our problem if we force our feature extractor to compete with a discriminator. This discriminator wants to maximize its accuracy of figuring out if a sample is cell line or patient. At the same time, the feature extractor wants to minimize its error to be caught by this discriminator if a sample is cell line or patient. What happens in the feature space that if they were not aligned, cell lines and patient, adding this discriminator helps us to get such a hidden representation that now cell lines and patients are aligned and all the responders and non-responders are on the correct side of the decision boundary that this multitask part is doing for us. In a way, the discriminator pushes feature extractor to extract invariant features from cell lines and patients, mapping them to the feature space that are very similar to each other. And at the same time, feature extractor is trying to do that by minimizing the error. And we have the multitask part, so we know that we are making accurate predictions during this process. So if I put everything together, this figure may seem a little bit scary, but I'll walk you through it. At the input, we have gene expressions of cell lines and patients as input. We have a feature extractor that extract features from both domains. I'll also do some math down here for folks who are interested. We extract features from both of them using a feature extractor, use those extracted features in a multitask subnetwork that has a shared part for both cell lines and patients, and two specific parts for classification for patients and regression for cell lines. And we optimize this network simultaneously for cell lines and patients to make sure that we have accurate predictions for both of them. And as I said before, we can apply the classification part to cell lines to get categorical predictions for them. And we can apply the regression part on patients to get continuous prediction for them, which, as I mentioned before, solves our output space problem. We also introduce a set of discriminators to use the idea of adversarial learning to align the input space problem. The global discriminator is just like the discriminator that I described in the previous slide. It takes the extracted features of cell lines and patient and shouldn't be able to discriminate them accurately. We want the feature extractor to extract so close feature for cell lines and patients to fool this discriminator. So to help team discriminator, we introduced a new set of discriminators called class-wise. The class-wise discriminator follows the same idea, but takes samples only from a specific class. In the simplest case, if we have responder class and non-responder class, the responder discriminator takes only responder samples and shouldn't be able to figure out if this responder sample is a cell line or patient. Similarly, the non-responder discriminator takes extracted features from cell lines and patient and shouldn't be able to figure out if this non-responder sample is cell lines and patients. And putting this all together, we can have a deep neural network that predicts accurate drug response using this multitask network and respects the discrepancy that we have in gene expression side and align it using the discriminators. And because as I said, drug response prediction is a case of inductive transfer learning, and we are using adversarial learning, we named our method ATOL. You may have different pronunciation for this. At some point, we have 9 people in our lab and 10 ways of pronouncing this, so pick yours. So we went through all this trouble, and the most rational thing is to ask if this is going to improve anything compared to those methods that they can't do any transfer learning or they don't want to do, or compared to those methods that can only adapt one side of the story, either the input side or the output side. And we also wanted to see if there is any meaning behind these predictions with respect to known targets of the drugs that we want to make prediction for. To train our model, we use GDSC cell lines, the panel of 1,000 cell lines screened with 265 drugs. And we use different sets of clinical and preclinical data sets, patients and PDXs with drug response as target. Based on the availability of number of samples, we chose these 4 drugs on the right side to build our models for. And we also gathered TCGA patients without any drug response to perform another step of test. So here we see that in terms of area under ROC, our method ATOL, the last row, outperforms those methods that they can't do any adaptation, methods that can only do input adaptation, and one example of a method that can only do output adaptation. And this confirms our hypothesis that adapting both input and output improves the prediction performance compared to ignoring the discrepancy or respecting only one side of the story for drug response prediction. We also performed an ablationist study to see the impact of different components that we designed for ATOL by removing, for example, both adversarial parts, removing only global discriminator, and removing the class-wise discriminator. And we saw that still ATOL in the complete format, when it uses all these components and stop networks, can outperform its own variants, which shows that we need to use both multitask part and adversarial part in the global and class-wise level if we want accurate drug response prediction. We observed similar pattern for area under precision recall. Here, ATOL in the green bar at the end outperform all the baseline for the majority of the drugs, and also compared to the random classifier performance, the horizontal red line, which shows that in terms of both AUROC and AUPR, our method is outperforming the existing state of the art. The last study, we wanted to see if there is any meaning behind the predictions that our model is making. So we used trained ATOL and apply it to different TCGA cohorts separately. We selected the target genes, those genes that we know these drugs were designed to target or had known association. We selected this list from PharmacoDB database. And we simply fit a linear model between the level of expression of those target genes and the responses that our model predicted. And we observed significant associations in different cancer types across the target genes. And this simply tells us that the responses that we are predicting, they're not some random numbers, and they mean something with respect to the target genes, those genes that those drugs were designed or known association with, which was a good sanity check for us. And we followed some existing works on this to perform this type of analysis. And it showed that the predictions, they actually mean something. So to summarize what we did in ATOL, we proposed the first method that can perform both input and output adaptation using multitask learning and adversarial learning. And we showed that that works. And if we want to have accurate prediction, we need to respect both sides. We published this paper last year at Bioinformatics. If you want to read more details, please check the paper, and all the codes and supplementary material are available on our GitHub repository. But there's a problem here. ATOL requires labeled target domain, patient data, or PDX data for training. I told you right at the beginning that these data sets are often small. And even though we are using multitasking to reduce the need for have a super large data set, still having a small sample size can reduce your generalization capability. And this is a huge burden for us. More importantly, having access to this labeled target domain is a big assumption in the first place. Ultimately, these models should work during the deployment time in a clinic. And when a patient walks into a clinic, we don't know anything about response to a particular drug. We don't even have access to that patient. And we cannot retrain these models every time a single patient walks in and we want to see the performance. So we need to be able to have methods that can take unlabeled data and doesn't require retraining every time that we see a new patient, which is what we are currently focusing on to solve for ATOL. But I want to finish with some open questions, some other areas that are very important for drug response prediction, some of the challenges that I mentioned at the beginning. And then I have some thoughts for computational folks in the audience. So we know that gene expression is the most effective data type, but there are recent studies that they show the utility of other omics data type. But when you have multiple omics data type, the question is, how do you want to integrate them? Broadly speaking, you have two approaches, early integration or late integration. In early integration, you literally concatenate different data type and data set, create one giant combined data set, do your computational modeling, and then your downstream analysis. But this ignores the unique characteristics of each data type. For example, mutation is generally a binary matrix, whether a gene is mutated or not, but gene expression is a continuous value. Plus, when you're concatenating these data sets together, you're increasing the dimensionality, which, as you know, is a huge problem for any of these data types separately in the first place. So late integration tries to solve these problems by performing computational analysis on each data type separately, and then finding a way to integrate these obtained results in the downstream side. So we proposed a method of late integration using deep neural networks that combine mutation, copy number, and gene expression. I don't want to go into the details of this computational tool that we developed, but the message here is that we showed that late integration in green can outperform early integration in terms of AUROC, and also can outperform gene expression, the most common data type for building these predictors. But what I want to say here is that integrating these three basic data types is not enough, and we need to go and move beyond these only mutation, copy number, and gene expression. Two famous and well-adjusted examples are proteomics and germline mutation. We know that protein is closer to the phenotype compared to gene expression. And there are some evidence that they show the utility of proteomics data for accurate drug response prediction. But unfortunately, current resources lack proteomics data. Only one or two of them provide this type of data sets, and sometimes they're even very small. Plus, unlike gene expression, a standardized tool doesn't exist for proteomics data. So we need to find a way to gather more proteomics data and use a standardized tool that can handle these noisy data types, because they tell part of a story that we can get using gene expression or other omics data type. And it's a similar story for germline mutation. If we want to have unbiased analysis, we need to look into germline in addition to somatic mutation. This study, for example, showed the utility of germline mutation in addition to somatic mutation for getting more accurate drug response prediction. And they also showed that germline captures something beyond gene expression. So we need to look at all these different data types. If we want to have successful methods, multiomics, in my opinion, is a way to go, but not just focusing on a handful of omics data type. We should be able to look at different omics data type, as many as we can get. But only making accurate prediction using different data types is not enough. We need to move away from treating these models as black boxes. We should be able to explain why we are making certain prediction. And understanding an accurate model can help us to gain new insight from the disease or the way specific drugs may work. One example of this effort is the BDCAN model that we designed last year, a knowledge-based neural network that we curated the architecture of this network using known biological knowledge. We designed a gene layer, a protein layer, pathway layer, and drug layer, and connected them together based on domain expert knowledge and known biological knowledge that we know that genes forms protein complexes are part of pathways, and drugs are generally designed to target specific pathways. And we connected them that we know from the biology this link exists using these green connections. But we also provided a network with black connections, those connections that can exist, but they're not supported by biology. And when we trained this whole system, we forced the unsupported connections, the black one, to die and become zero, with the hope that at the end, if you're having an accurate model with some black connection, go back and study those black connections, because there's something that we don't know based on the biology, based on the domain expert knowledge, but they survived the accurate prediction process, which means that probably they contributed something for accurate drug response prediction. And we showed across 4 drugs that this BDCAN version, we call it BDCAN Plus, that use both green and black connections, outperform the version of BDCAN that only has the knowledge base, the green connection, and 2 other networks that have the same architecture or they use some fine-tuned architecture in terms of RMSE. The beauty of such a knowledge-based network is that we can query it for a specific drug. This is an example of important subnetwork that we extracted for tamoxifen, for example. You see that each layer, the most important entities that contributed and the amount of contribution based on the thickness of this line. Immediately, you see that, for example, tissue type 17 was very, very important for making accurate prediction. And when we go back and check, we found that this was indeed the breast tissue type. And you know better than me that tamoxifen is the primary treatment for breast cancer. We also observed different genes that all had known association with breast cancer. So it shows that when we use these knowledge-based networks, we are making accurate prediction, and we can ask interesting questions that what drives these predictions. I don't want to get into the details of every of these entities, but there are some unknown connections here that that might be worthy of wet lab experiment to see why they appeared in making accurate prediction for this particular drug. So finally, let me conclude with some computational thoughts. As I mentioned, we need these models to work at the deployment time when a single patient walks in the clinic and we don't know anything about that patient. And this was generally the figure that I explained for transfer learning, that we want to make accurate predictions for the data that we care about. But in the real world, in the clinic, this link is broken. We don't have access to the patient that may walk in the future, but yet we still want our models to make accurate predictions for them, using only the source domain, for example, the cell lines or previous patients that we had. Domain generalization is a family of computational tools in machine learning that can help us with that. They're also known as out-of-distribution generalization because their goal is to generalize to something outside the distribution of the source domains. We want to make accurate predictions for patients in the future while we don't have access to them during training. And there's a very, very active line of research in purely machine learning community and bioinformatics and computational biology community for applying domain generalization in different problems. So what I'm hoping that you get from this talk is that biases exist in different data sets. What I talked about was in the context of drug response prediction, but it can exist in any problem as soon as you move beyond your own data set. And even there can be the case that you have different subgroups and biases within your one single data set. And transfer learning is a family of methods that can help you to handle these discrepancies and ask interesting questions to your data. And you can integrate it with other areas such as multiomics, when you have multimodality data with multiple modality, or you can make it knowledge base to be able to interpret why you're making certain predictions. So at the end, I would like to thank people who were directly or indirectly involved to some of these studies that I talked about, different institutes and funding partners. And I would like to thank all of you for joining us today. If you need one more details, feel free to check some of the works that I described today. And always, you can reach out and we can have more targeted discussion. Thank you so much.
Thank you very much, Hossein, for the interesting talk. We now want to open the floor to questions from the audience. So if you have questions, please type your questions in the Q&A feature below. To kick off the question section, I have a question. I have many questions, but I'll start with one, which is on the multi-omic and the benefits that you mentioned. So as you also mentioned that one of the challenges is that as you increase the dimension, you have challenges in the sense of fitting the model. So do you believe that the late integration is the way to go with multiomics?
That's a great question. I think yes. There's also one intermediate integration family that I didn't go through, and to me, it's a subset of late integration. But I think late integration helps us to respect the unique characteristics of each data type instead of ignoring that as it happens in early integration. And this is why I didn't go through this method, but this is why we had separate subnetworks to extract features for each omics data type separately, because we need to be able to respect the extracted features of different, for example, from mutation is different than copy number or expression. At the end, we need to integrate that, but at the beginning, it's important to see them separately.
Yeah, that makes sense to me. Yeah, I agree.
It makes sense. I'll follow on. I like Chia have many questions, but I will, I'll pay attention to the Q&A, and as soon as people chime in, I'll stop asking my questions and ask other people's questions. But between this approach and BD-Con, which is using the knowledge bases to integrate that data, I mean, it seems like that Molly is going to be stronger in that it's going to be generic and it's going to allow you to take in many different types of data without necessarily understanding how they're interrelated. But BD-Con seems like it would be better at dealing with that dimensionality problem. Can you comment on that?
That's a great point, Chia. And yes, BD-Con and Molly may seem not related, but in fact, to me, the ideal situation is to models like BD-Con to always extract features from different omics data type, because we can go back and see why certain layers of information like genes, proteins, or pathways were involved in extracting that features. So in principle, we didn't try that, but in principle, we can have a mini BDCAN for each of these subnetworks, or even for ATOL, to extract features from them, and then be able to figure out why, which part was, drove that extracted features or accurate prediction. So I certainly see a benefit of integrating all of these into one model eventually.
Okay. Kia, you have, we'll take turns if you have more questions, Kia.
I'll jump in here since we're on the top of Multiomics, and Kia opened the floodgates to a Multiomics panel here. On the topic of Multiomics, Hossein, question for you is, this framework, Molly, BDCAN, have they, or did you develop new methods that allow you to overcome challenges associated with lack of standardization or class imbalance that we're naturally going to run into when you're integrating these different modalities?
Yes. For example, for Molly, I wish now I had more slides on Molly. For this method, we had a dual loss, a classification loss for accurate prediction, and a triplet loss to manage the embeddings. What a triplet loss does, it takes samples in the form of triplets of 2 responders and 2 non-responders, and it forces the integrated representation that responder samples cluster together and more distant from non-responders. Imagine even for a data set of 200, 300 samples, how many triplets you can create. And you can put constraint only focusing on the hardest examples or the easiest example. So immediately you move away from having imbalanced classes, and you can create indirectly more samples for yourself. And in addition to that, we can always go to more classic approaches, for example, oversampling the minority class while we are training, which may lead to overfeeding, but with a stronger valorization, we can control that. So we try the combination of these to help us in Molly and BDCAN to overcome the imbalanced classes. And that's why we are looking at both AUROC and area under precision recall, because precision recall is a way to go for evaluating the performance when you have imbalanced classes. I didn't present the AUPR results here, but you can check in the paper, and they follow the same pattern in terms of the performance of Molly.
So we do have our first question that's popped up on the chat window here, and we're going to flip you to the other side of the algorithm. What feature selection algorithms were used?
For
any of them.
We didn't use any classic feature selection approach per se. First step is to take those features that are in common if you have multiomics, or in terms of, for example, ATOL, if you have gene expression in cell lines and patients, we focus on the genes in common with them. But we usually have a very intermediate, very naive filtering step that we get rid of the genes that they have very low variance, because it's like having a number of, for example, feathers for birds as a feature. It's 2 for all of them. It's not going to help any model. So if a variance is low for a feature, we removed it. But we didn't do any classic feature selection on any of these methods.
So no initial dimensionality reduction step.
No.
Feature extractor. I think that answers the question. Kia, do you want to?
Sure. While we're on this page, so there are 2 hyperparameters here, right? Lambda G and lambda DC, which I'm assuming when it comes to like adversarial training, these parameters are important. Did you do hyperparameter optimization? Is the method stable with respect to them?
Of course, I didn't go into detail, but hyperparameter tuning is always there when you go to these deep neural networks. And we performed k-fold cross-validation, I think 5-fold on this part of the data for tuning hyperparameters and also our cell lines. And I don't quite remember in terms of the stability of the values, but in the ranges that we tried, it wasn't that difficult to find the best value.
Another question, I guess, while we're on the same topic. What sort of loss did you use for your discriminator? Was it any sort of like distributional?
The loss is a general binary cross entropy, because the way these discriminators work is 0 and 1 label. For example, 0 is cell line, 1 is patient. And when they receive a sample extracted features, they treat it like a classification problem of accurately classifying these discriminators into the proper classes.
So there's another question that's popped up now, and I think I have a very related question to this, so I'll combine it. The question is ADL used for early integration? Which omics layer did you use in this method? And I guess related to that question is my question, which is can you discuss some more about how the performance scales, both with the number of samples, the type of data, and the combined modalities? Does that make sense? Because I think the practical question that the biologists want to know is, how much data do I need? Have I already achieved my goal, or am I never going to achieve it with ADL, or is it somewhere in between?
Well, we achieved it, so it's not impossible. And in terms of modality, we only focus on gene expression, because getting multiomics data for patients, clinical trial patients, it's not easy at all. So we only focus on gene expression in terms of modality. And in a sense, there was no early or late integration for that, because they had a shared feature extractor, but they went through it separately in this model. We did not concatenate them or combine them. We always respect that these are two separate samples. And that's why when we were training, our issue was that we had, at the beginning, we had separate embeddings in the feature space.
So how large a data set did you use?
So in our data sets,
how many cell lines? And
yes, GDSC generally has roughly around 1,000 cell lines.
And those were all different unique cell line experiments?
Some of them are the same cell lines, but some of them are unique, some of them are replicates. But the good news is that we used GDSC V1 in this paper last year, but now we have access to way more cell line data sets. Just at what I presented at the beginning, we have CTRP, which is close to 800, GCSI has 700. So if you need more cell lines, we have that. In terms of patients, we are a little bit limited, and I think for each of these drugs, we could find roughly around maybe 100 patients or PDX samples, no more than that. And that's why that was the criticism that I had for ATL, that 100 samples may not be enough for generalization to future patients. And ideally, what I would like to see is that we get rid of this part. We shouldn't be relying on labeled clinical data, but instead, if we can take, for example, unlabeled data from TCGA or any other big consortium, if we can use them instead of the labeled clinical trial data, it reduces a lot of, it removes a lot of problem that we have in building these models.
And I guess, there's a trade-off in my mind, and I'm gonna just, I'll give up. I'll stop talking after this because I'm taking a lot of time. But there's a sort of trade-off in my mind between having more experiments in the same circumstances, having heterogeneous experiments, different cell lines, and having different experiments. And so I was wondering if you tested, how well the transfer learning, the dual prediction layer worked when you're using the cell line data and the clinical data, when you took out certain parts of the cell data. I mean, I was wondering if you use that as a way of understanding what is the most important aspect of that.
In the Appalachian study, we took away different components one at a time, but we did that using the patient data. We never withhold the cell line data because obviously our goal was to accurate predictions on patients. Right. And that's why we withhold some part of the patient data for tests. And for us, cell line was a way to achieve that, but never the ultimate goal to see if you are perfectly accurate on any other cell line data sets. So I think the answer to your question is that we use all the cell lines that we could get, but we use a portion of the patients to see how well it works.
And I'm kind of curious if I guess you haven't done it yet, so you've answered the question, but I'm kind of curious, you know, how much cell line data we need to create the perfect, you know, predictor.
I think, let me go way back at the beginning. All the models that I described, I'll present it here in this overall figure. You see that the majority of them use the same 1000 GDSC cell line. And to more or less, they all showed some levels of utility in making accurate predictions. But as we get more and more cell lines or patients or drug interaction or adverse drug reaction data sets, people are finding more innovative ways to merge different data sets. For example, you see that now there are studies based on multiple cell lines at the training side, as well as some drug information next to it. There are studies based on patients and PDXs, and many other combinations. So the more we get, I think the more we can act on some of the challenges that we have, some of them that I described at the beginning. But in worst case scenario, for example, in 2014, a simple ridge regression using only GDSC, or back then it was named CGP, can also make accurate prediction on patients. But the more you have, the more validation you can do, and more interesting questions that you can ask.
Okay, thank you very much.
A quick question, just because I see a lot of oncologists on the attendee list. One question that I'm sure I would get, and I'm most likely going to get if you don't get it first in your email box, is, have you explored the effect of multiple drug combinations, i.e. data from multiple drugs as opposed to just monotherapies? How well would ADL perform?
We did not explore drug combination. I had some slides for open question for that, but I was going way over time, and so I removed that part. We didn't go on drug combination, but certainly we know that one drug does not cure cancer. And luckily, we have standardized resources for synergy between different drugs. And obviously, extending any of these models to predict, to make predictions for multiple drugs, the combination of the drugs is going to provide us with more calibrated and more usable methods for the clinic.
Makes sense. So final question is, have you observed a difference in ADL's performance when predicting response for targeted therapies versus cytotoxic agents? Question from the audience.
Well, that's a great question. We specifically picked drugs to target narrow impact chemotherapy and broader targeted drugs. Based on my experience, some drugs are a little bit difficult. For example, paclitaxel is a very toxic drug and kills everything immediately. But we generally observe a little bit better performance when it's targeted for these models. But it depends a lot on the type of drug that you're using and the amount of samples that you have for that drug. So we can get both sides, that we make accurate predictions for both chemotherapy or targeted drugs. But based on my experience, it's a little bit, things are a little bit nicer when it's targeted drugs.
Makes sense. So we're approaching the top of the hour, and I want to thank Mr. Sharifi Noghabi for joining us today and providing a fantastic presentation on transfer learning and drug response prediction. I think ADL and a lot of your work in future directions holds a lot of promise for clinical deployment. So if anyone in attendance has any lingering questions, please feel free to reach out to anyone on the panel, including Hossein, for any follow-up questions. And please stay tuned for future talk by Dr. Yuri Shalit on June 15th and other upcoming talks in this AI speaker series. So once again, thank you everyone for joining us today. Take care.
Thank you.